to Gebze Technical University, Department of Bioengineering webinar series. Our guest for today's webinar is Dr. Tuğba TOPAL from Uşak University, and the webinar title is Bioengineering Approaches to Force Stem Cells to Behave on Cellular Microenvironment. Dr. Tuğba TOPAL was completed her bachelor degree at Marmara University, Faculty of Arts and Sciences in 2009. She completed her master and PhD program at University of Michigan, Biomedical Engineering. During postdoctoral studies, she worked at University of Michigan, Department of Chemical Engineering, and the National Institute of Health. Currently, Dr. TOPAL have been working as academician at Uşak University. Dear doctor, stage is yours.
Sweet. Thank you for the introduction. I hope my voice is clear.
Yes, it is clear.
Okay, thank you for a kind introduction again, and thank you for having me for this webinar. Today, I will be talking about the forcing stem cells to behave by using bioengineering approaches as, as our host mentioned. All right, I don't know what's happening. Okay, the overview and goal for this project, I mean, not this project, the projects that I'm going to talk about, is basically establishing bioengineering approaches that are used to differentiate, maintain, and support human embryonic and pluripotent stem cells to study biomaterial interaction that drive changes in tissue behavior. To do that, our goal to match the functional complexity found in living system to control or restore biological function, and our methodology is to combine chemistry, material sciences, engineering approaches to achieve control of these properties and multiple length scales. So as we all know, all the cells in the body is coming from one cell type called zygote, and it's called zygote. You can make them from red blood cells, nerve cells, and tissue cells, and whatever you can think about it. So this is a time lapse from a mouse embryo, this in pre-implantation development. As you can see, one cell becomes 2, then 4, it increases 2 to n manner, like exponentially. And at some point, and it's like the embryo, the egg starts hatching and it's going to the uterus. But we're gonna talk about most of the part of it at the first part of the talk about like early development, and later on I'm gonna talk about the different methodologies. So to brief, I just wanted to draw your attention to human embryonic stem cells or pluripotent stem cells. We know that they have ability to self-renew and maintain pluripotency, meaning self-renew means like they can make their copies of themselves without differentiating for a prolonged period, and pluripotency means they can make like 3 different germ layers, ectoderm, mesoderm, endoderm, means like you can make like neuron cells or bone cells and whatnot. So I just want you to pay attention to these pluripotency markers, because I'm going to talk about these 3 most of the time in my talk. Non-op04 and SOX2, these pluripotency markers are the core regulators of to maintain pluripotency of hESCs and iPSCs. If you see any of those lost from the nucleus, it means that these cells are differentiating. If 3 of them maintain, if there are 3 of them is just like localized in the nucleus, they mean they are undifferentiated. So what are these cells? What are we going to do with them? So we can use the cells to use in regenerative medicine, means you can use them for cell replacement therapy, like using for Parkinson's and Alzheimer's, or we can use these for drug testing, like identifying drug targets or testing potential therapeutics. So researchers so far, they've been using different strategies, like, for example, biophysical cues, extracellular matrices, or chemical stimuli. Basically, there are like a lot of like approaches to control it. So in the first part, I will be talking about biophysical cues, mainly mechanical strain, applying forces to the hESCs to differentiate them. So, sorry. And researchers in the field, especially engineers, they are able to use different forces to understand how these forces altering cell fate or understanding the understanding forces in the body. In this case, we know that there's like stretching in the body or compression. You can think your teeth, or you can think like shear stress in your blood vessels, because they become most likely like a flat-ish shape. Due to all these forces, the cells, they change their there's a term called tensegrity, or mechanical biology part of it, by Donald Ingber. So he basically introduced this term back in the day, saying that due to these like forces applying to cells, the tensegrity of this like cell, that it changes, the force changes the cell behavior. In this case, you apply force. In this case, like for example, mechanical stretch, you apply this force to the inner greens are getting it, and then focal adhesion kinase are activating, and then cytoskeleton and changing nucleus, gene expression protection, blah blah blah. So this is just like overview. So there are like forces in the field, especially engineers has been using. There's one called atomic microscopy. You can use this up to 100 nanonewton, or you can use magnetic tweezing cytometery. Basically, you introduce a bead and you can move this bead through the tweezing cytometery. And there's also called nanotopography, ECM nanotopography. Basically, you make this surface stiff or soft and understand how the cells are behaving. Or you can use vacuum-based, the mechanical strain. This is Flexcell. It's a company that you get basically using vacuum and applying strain from two sides, and basically applying force to cells and understanding how they react. So how do physical forces alter stem cell fate during development? So the principal hypothesis is coming from, as I mentioned earlier, fertilized cells become like 2-celled, and 4, 8, 16. Again, it goes 2 to n manner exponentially. And at some point, there's no growth factors, or there's no there's nothing coming from outside to inside, and nothing getting inside outside. Basically, everything happens in the egg. Like you can think the egg like you eat daily. So when they reach the 16 cell, they start before that they start differentiating. And the principal hypothesis, when I was like working my PhD, just like, oh, there must be a lot of compression forces, or low cell contact and surface tension and compression forces. These are the forces that are starting these cells to differentiate, because before hatching, these cells, they know what to become next. Like for example, purple, the color I put as like a purple, these are gonna make the placenta. How do they know? Or these and then these gray and the yellow, they like the hESCs are coming from the very temporary from this side, and there's like the early endoderm layer in this area. But how do they know what to become next? So to test this hypothesis, we wanted to assess the effect of integer-mediated cyclic strain on hESCs, and to do that, we established a protocol or a technique I will be referring as ATC, acoustic tweezing cytometery. Basically, we use a lipid-shelled microbubbles coated with RGD peptide targeting alpha V beta 3, and apply 45-degree angle and apply ultrasound to the cell for 17 to 25 nanonewton. And we apply for 30 minutes ultrasound application, as you can see. This is basically the technique that I will be talking about the first part of the talk. So as you can see here, we first applied these forces to single cell level and colony level, and we wanted to see how they react. As you can see, these black dots there, or microbubbles. And as you can see here too, it's in the colony, edge of the colony. And when we give these bubbles to these cells, they prefer sticking on the periphery of the colony, not in the middle. So the next, I just want you to pay attention that how we do it is just like, again, this is like single cell colony, and we apply for 50 milliseconds and 10 milliseconds of duration, and we apply for 1 second for that, and then we do it just like for 30 minutes, and we apply for, stop it, apply again, stop it, so vice versa. All right. Do you guys still see me? I'm not sure because there was a thing here. It's disappeared. You guys hear me? Hello?
We can see your slides and hear your voice.
Okay, sweet. All right. So since we are working with the human embryonic stem cell, the next question before that, we wanted to see, we worked how the contractility changes. Contractility means how forces, when you apply the ultrasound forces, how it's going to happen, the contractility. So for this experiment, we use micropillars, as you can see here, the pillars. So we did this with the jumping phase group at the University of Michigan Mechanical Engineering. We put this with the micropillars, we put the cells on it, and then this is like no bubble, no ultrasound, and this is bubble and ultrasound. At the beginning, you don't see, the red color means there are a lot of contractility, blue it means there's nothing. As you can see here at the beginning, there's nothing, and then like nothing, nothing. It's just like not like perfectly blue, but I want you to pay attention here that after 30 minutes, we apply forces, and like I mentioned earlier, the bubbles are like located at the periphery, but when you apply forces, the contractility accumulate in the center of the colony. And we tested this with the normalized force, and we applied an inhibitor called Y27. It's basically a ROCK inhibitor. And we tested this whether if we apply the ROCK inhibitor, whether contractility happen or not. And when you apply to inhibitor, we were not able to get the contractility as without the the inhibitor. All right, so since we work with the hESCs, the next question is just like, okay, this is human embryonic stem cells or pluripotent stem cells. Have you checked the nanographer and Saxto? And yes, we did. And as you can see here, the single cell level and the colony level, at the single cell level, you can see nanog and upward disappeared, and Saxto expression is still there. And we did the same thing for the colony level, applying 30 minutes of forces, and nanog and upward disappeared, Saxto expression is high. And this is like only the confocal analyses, the image technique. And then we wanted to correlate with the protein expression analyses. And we did the upward Saxto nanog again, with and without microbubble and ultrasound. And as you can see here, there's like after 30 minutes, upward nanog disappears and Saxto overexpressed. And we were just testing and fishing multiple antibodies, and we wanted to see whether, since this is like early differentiation, whether we can do like EMT. We wanted to test vimentin, and then we've seen vimentin expression is high, means these cells are going to epithelial-to-mesenchymal transition. And this is very well known in the field. Biologists, they were able to figure that out. Like if these cells going to primary EMT, and before they make a neural crest elimination, they are going through epithelial-to-mesenchymal transition. So it's also another paper about talking about if the E-cadherin, beta-catenin, and alpha-catenin, blah, blah, they go down, means these cells and the vimentin and cadherin fibronectin goes up, it means that these cells are going to EMT transition. So we tested this. Before that, I want you to show that activation of focal adhesion kinase. This is very important in the field of the human embryonic stem cells because my colleague, he was able to use this activation of focal adhesion kinase using manganese treatment for 48 hours. And he was able to see activation of FAC and basically triggering human embryonic stem cell to differentiate. So we tested the same hypotheses with the ultrasound technique, the ATC, and we were able to get phosphorylation of FAC localized in the one part of the cell. And at the same time, since we were able to see the vimentin increase, and we were like, okay, what's going to happen with E-cadherin and cadherin? And yes, E-cadherin is highly expressed in human embryonic stem cells, and when you apply the forces, E-cadherin is lost. So these cells are definitely going to epithelial-to-mesenchymal transition. And we further tested whether it's true, and yes, we did the same thing for N-cadherin. And we further correlated with slug and snail because those are the early markers of rapid EMT induction. So the next thing is like biologists, they were able to figure that out, how to make neural rosettes and neural differentiation. They were on 2D, they were able to do PAX6X1 expression. These are early neural ectoderm determinants. they were able to get this from at day 14. They named them as a rosettes, or we can call them roses, whatever you want to see it. But they are so smart, they were able to figure this out like early, or at day 15, and they call them. And then we wanted to test this hypothesis with our technique, whether we can decrease that time point. Means they were able to do for 14 days, 2 weeks basically, whether we can just put it down and whether we can really mimic early embryo. On 2D though, so we did exact same technique. We applied the force, and then after 30 minutes, we gave them neural induction media, and then we continued for up to 48 hours. At 6 hours, we started seeing PAX6 and SAX1 expression, and 48 hours, we started seeing PAX6 and SAX1 expression and rosette. So as you can see here, this is early determinant of upregulation of PAX6 and SAX1 in 6 hours. And later on, bam, here. So we get the rosettes after 48 hours. So, and then we wanted to test our hypotheses again with the inhibitors. So we did like, what if we block myosin 2 with the blobstatin or focal adhesion kinase inhibitor, and again, ROCK inhibitor. And as you can see here, we did not get any of those markers upregulated once we gave them the inhibitor, because they basically inhibit the pathway. So our conclusion from the first part is like basically cyclic forces generate rapid mechanical responses in ESCs, and it can be utilized as a new technology to improve cell fate determination and for regenerative medicine. And cyclic forces induce increased cellular contractility and differentiation of ESCs as fast as 30 minutes, using these techniques to rapidly generate high-quality neural differentiation to test in vivo. So the next part I will be talking about the extracellular matrix part of the controlling stem cell fate. Basically, I will be talking about synthetic materials, that how we can alter stem cell fate on it. So to do that, like I mentioned earlier, ESC, I did not talk about how we culture it. I just talk about how they react, where they are coming from and whatnot. So ESC culture, or pluripotent stem cells, they need 2 things, a matrix and growth factors. So cells can grow in colonies, and they can transfer into many cell types. So there are challenges to maintain pluripotency, and because they have irregular shape, they make irregular, I mean, also you make it in 3D, they make irregular shape in colonies, embryo bodies, and they form cell dense tissues, and they start differentiating spontaneously. So this is how they look, how ESC is looking like as a clump or colony. And like I mentioned earlier, if you see them in the nucleus, it means they're undifferentiated. Can grow in colonies, and they can transfer into many cell types. Stem cell renewal. This is the study that we did at the Germany. Karlsruhe Institute of Technology. Basically, it's a polymer, zygote. It has plus and minus ends. We use this polymer because before me in my lab and Jörg Lahan's lab, they were able to figure out, and they published that in Nature Biotech paper about a polymer, that a polymer that supports human embryonic stem cells self-renewal. So we altered a little bit of this polymer, and we investigated the same way whether we can maintain pluripotency on this polymer. And yeah, so this is the German part of it, how they did it, using the CVD coding, substrate, and using the linker, using microcontact printing and whatnot. And later on, Gauthami did this work part of it. She was able to use the protein, whether we can put the protein and whether they are gonna absorb the polymer or not. So, and then the next thing we want, since we wanted to test whether we can maintain stem cells on it, and or control group, of course, Matrigel, because this is like everybody used the Matrigel. At the beginning of the passage 1, we get like teeny colonies, and later they get used to it at passage 5. And we correlated, they were able to maintain pluripotency without differentiating, and we correlated with the Western blot analyses, as you can see here. the beta-actin is a control group, and these are the pluripotent marker. This is a naive marker. After 5 passages, and as you can see, they're still like highly maintained their pluripotency. And this is qPCR analysis. It means there is no changes in the gene expression. So our conclusion from this part of the work is using polymer surface represent a significant step in the development of fully defined reproducible culture system for ESC for cell expansion. So the next part is how do 3D cell culture approach maintain stem cell self-renewal and support direct differentiation. So basically using those 3 approaches in one. So we all know the 3D cell culture approaches, and there are like limitations. Basically using a lot of synthetic materials is not, especially if you're using for regenerative medicine purposes, it's no good. So we should minimize the interface for these materials. And also there are problems about limitations about transport properties and lack of hierarchical control. And because like you make it, but you cannot control the way it is. And other challenges is like 3D culture of undifferentiated stem cells. They are like forming embryo bodies, uncontrolled or heterogeneous or limited in size. And researchers have been using nanofibrous mats, sprays, or hydrogels to make 3D cell cultures. So in that case, this is Jake's paper. He published this in 2016. It's basically a 3D jet writing technology. Sorry, I said 3D jet writing technology. Basically, you can make like squares, rectangles, or orthogonal, or whatever you want. It's like it's basically a scaffold. You can print the size or the color. I mean, not the color, the size or the shape, whatever you want with this 3D printing. So first, in his part of he wanted to test this with using this material using mesenchymal stem cells, and comparing with the plastic that 2D and novel and PLGA made on the material too. And then he did this with just to show whether we can coat with fibronectin as the extracellular matrix and whether we can culture mesenchymal stem cells in it, and he was able to do it. So here we use this technology just for to provide mechanical support for matrices that maintain pluripotency, directing differentiation, and supporting neural growth. We can also use this technology to vary material substrate parameters that influence neuron behavior. So how we do it? This is the part of the printing part of it. And then later we do the extracellular matrix coding, and then we see the human pluripotent stem cells on these. Like I mentioned earlier, these pluripotent stem cells, they can make any kind of cells. In this part of the project that I'm going to talk about, we mainly focused on the neural part of it. So characterization, I will be showing the images and the single cell sequencing, not really in this case. All right, so how we do it? So we do the 3D jet writing, as you can see here, and then cut them into little pieces and put it in a metal frame, and doing the fibronectin coating for 2 hours, because these cells, they cannot just sit on a PLGA, so they need some cushion to sit on it. And then we gave them like pluripotent stem cells, and then at day 15, they maintain pluripotency and whatnot. So here are the data. So after 15 days, you can see they've completely fully covered the entire scaffold, and we were able to maintain the pluripotency and self-renewal on these. So, and the next is whether we can differentiate the cells using neural induction and maturation media on these. So to do that, again, at the beginning, we seed the cells, we give them neural induction, and we start seeing beautiful rosettes, like I showed you earlier in our ATC technique. And then after 21 days, we get a neural at the beginning midbrain differentiation. These are all commercially available media, by the way. And then we give them midbrain neural maturation. So as you can see here, they're like, this is 2D. There's no, after 7 days, there's no rosettes, but at the 7 days, we get the neural rosettes, and later on, I will be showing. This is the 3D images of it. And this is the Western blot analysis of showing, yes, we are able to get this protein expression without differentiation. Oh, sorry, with the neural, with with the neural differentiation, this is the part that we show there is no differentiation for non-organic adherence there. All right, so what do we do here? This is we call them EECM. They supporting human pluripotent stem cell culture neural differentiation. So at the beginning, like I mentioned earlier, we make the neural rosettes, beautiful rosettes. They're appearing here, and then we further investigated whether we can make a fluid neurons, and you can see from here, maybe here too, better images of these neurons starting making it after 20-25 days. All right, so later, the PhD student, she further investigated to expanding maturation up to 7 months, and we get like a bulk of it, and we call them like EECM organoids. And she get a lot of single cell analyses of that. I'm not showing it because the paper submitted, so I just wanted to show the images part of it. So, but she was able to get like all these different part of differentiation and neural system and homeostasis, gene expression of transcription, blah blah blah. So with that, I hope I was able to show you, all of you, how biophysical cues is changing stem cell fate, and using SSM metrics, how it's changing the stem cells or maintain pluripotency, and using all of these in the 3D cell culture. So as a conclusion, we can do it is like bioengineering approaches to control stem cell fate using cyclic forces can be utilized a new technology. And we can use the polymer surface instead of using matrigel, because as you know, with the pandemic, there's a matrigel shortage. I'm not sure whether you're aware of it, but now we need some polymer surfaces in order to supply. And at the same time, we don't need xenogenic material that introduce ourselves if you want these in our body. And another thing is the last part is a 3D cell culture. We can use this to use as an array in the biotechnology application. The idea when I started this 3D culture, the material, since it's PLGA, it's biodegradable, I wanted to use this as a like getting the patient cells, getting it, making pluripotent, and differentiating it to neuron, in this case dopaminergic, and putting it back to the patient. So future directions can be exploiting mechanics to control post-transplant cell fate. For example, they may be valuable in building human-based in vitro models of organism diseases. As you know, a lot of people and researchers are working on these. And we can also bridge the gap between basic research and mechanobiology and improve therapies for patients. So with that, I would like to thank my PI, Paul Krebsbach. Now he's the Dean of the School of Dentistry at the UCLA. And of course, my mentor and another mentor at Auckland University, Louisville. And Joe, my buddy, and now he's an NIH NCI, of course, Fu Lab, Deng Lab, Sherry Deng, my professor, and Jumping Fu and Shufang and Takayama Lab. Oh, I didn't talk about that part of the project with the microfluidics. I thought I wouldn't be able to finish it. And other thing is Jörg Lahan's group. Here's Jörg Lahan and all my friends. And with that, yeah, so if you have any question, I would like to take it. Thank you.
Dear doctor, thank you for your impressive presentation.
Thank you.
Dear participants, if there is any questions about the presentation, please ask right now, and you could raise your hand to ask.
Merhaba hocam. Ben öncelikle çok teşekkür etmek istiyorum. Bizim çalışma alanlarımızla da ilgili çok güzel bir proje, birkaç proje bize sundunuz. Çok teşekkür ederiz. Şu an sanırım Türkiye'desiniz. Türkiye'de mi devam edeceksiniz?
Evet.
Hocam yani eğer çalışma alanlarınız bu şekilde devam edecekse altyapınızı düzenleyebildiniz mi ya da altyapı arayışınız var mı? Çünkü bunlar oldukça yüksek teknolojide gerektirebilen veya sarf malzemeleri masraflı olabilen, Türkiye'de bizi zorlayan teknikler açıkçası.
Çok güzel bir soru. Türkçem devam edeyim, İngilizcem bilemedim.
Devam edin hocam. Sanıyorum sadece bir Türkçe konuşmama ihtimali olan katılımcımız var. Buyurun.
Ha, öyle mi? Tamam o zaman. Çok doğru yere parmak bastınız. Bunların yani Türkiye'de düşünemiyorum bile. Hele ki single cell analiz tarafını hani bahsetmeye hani bilmiyorum. burada 10x genomics falan oturup onu alıp analizlerini yapmak tabii çok acayip masraflı. Dediğiniz gibi bu 3D printing alıp bunu hani PLC'ye de basıp ki ben pluripotent kök hücreden falan bahsediyoruz. Çok pahalı. Çok zor. Hani kolay değil ama düzgün yani ben şu an Uşak'ta bunu yapabilecek bir altyapım maalesef yok. Dolayısıyla yani altyapısı olacak farklı üniversitede collaboration yaparak hani mesela diyelim bir materyalci varsa hani materyal tarafını ile yapıp işte kök hücre tarafını başka bir hani beraber bir insanlar bir araya getirip amaç collaboration'ı yüksek yani yüksek tutup bir şeyler yapmak yani buna benzer şeyler ya buna benzer dedim bunu zaten yaptık ama hep hani pluripotent kök hücre daha çok regenerative medicine üzerinden gitmeyi düşündüğüm için o alanda devam etmeyi düşünüyorum. Bunu düzgün hani collaboration'larla yaparak tabii ki. Cevap verebildim mi sorunuza bilmiyorum ama.
Evet hocam yani açıkçası Türkiye şartlarında dediğiniz gibi aynı bu şekilde olması gerekiyor. Çünkü bir üniversitede işte kültür laboratuvarı var, öbürküsünde işte sekanslama var. Hepsini bir araya koymak neredeyse mümkün değil. Ama kolaboratif bir şekilde çalışırsanız aslında Türkiye'de parça parça altyapılar mevcut. Masrafı da artık TÜBİTAK'tan mı karşılarsınız? Yoksa Avrupa Birliği, NIH projeleri vasıtasıyla mı karşılarsınız? Zorlu bir yol. Yani Türkiye'de birazcık daha çaba göstermeniz gerekecektir. Herhalde siz de hissetmişsinizdir. Size iyi çalışmalar diliyorum. Yani Gebze Biyomühendislik'te birçok çalışma alanımız var ama ben biyoinformatik çalışıyorum kendim. Her zaman kolaborasyona açığım. İsterseniz bir buyurun gelin bizi ziyaret edin. Görüşelim, tanışalım.
Diyorum.
Bekleriz her zaman. Ben çok teşekkür ediyorum.
Ben teşekkür ederim. için.
Şu an başka sorum yok. Ağzınıza sağlık. Tekrar teşekkür ederiz.
Teşekkür ederim. Başka sorusu olan varsa alabilirim.
Hocam aslında benim bir sorum var.
Haydi sor.
Bu polimerlerin ve hidrojellerin boyutlarının ve stabilitesinin etkisi nedir acaba bu matrix olarak kullanımında, kök hücre çalışmalarında?
Yani degrade soruyorsun.
Sesinizi alamadım hocam. Tekrar eder misiniz?
Belli sıcaklıkta hani culture conditionlarında degrade olup olmadığını soruyorsan 7 aya kadar polimer orada durabildi, hani tutabildi. Sonra devam ettirmedik. Hani çok hani iyice büyüdü artık kocaman bir şey oldu. Ondan sonra hani ne olur bilmiyorum ama normalde PLGA materyali hani vücuda girdiği zaman kolayca absorbe edilir, vücuda da zarar vermeyen bir polimer. Ama culture conditionların yani culture durumlarında öyle bir şey rastlamadık. Çünkü vücut yani biliyorsun 37 degrees ama hani vücutta tabii daha farklı. Çünkü o sadece stationary olarak duruyordu ama vücutta mesela örnek vereceğim. Biz yine University of Michigan'da bizim bölümde ben başladığım zaman Scott Hollister'ın PLGA materyaliyle yaptığı trake vardı. Küçük çocuğa ekledikleri trakesiz doğmuş. Mesela o zamanla o PLG'yi orada yavaş yavaş eriyerek orayı önce bir scaffold sonradan orayı hani vücut kendi kendine oluşturmuş. O tamamen hani vücutta çünkü stationary olarak durmuyor. Orada bir sürü faktör var. Dolayısıyla cevap vermek gerekirse culture conditionlarında 7 aya kadar bir şey olmuyor. 7 aydan sonra bilmiyorum ne oluyor. Teşekkür ediyorum soru için.
Ben de teşekkür ederim hocam cevabınız için. Başka sorusu olan var mı acaba? Tuğba hocamıza. Sanırım yok. Tekrar çok teşekkür ederiz hocam. Değerli katılımınız için ve sunumunuz, verdiğiniz bilgiler için.
Ben teşekkür ediyorum davet için. Herhangi bir şey olursa e-mail'e iletişime geçebilirsiniz. Cevaplamaya çalışırım.
Çok sağ olun hocam.
Teşekkür ederim tekrardan.
Arkadaşlar webinarımız burada sonlanmıştır. Katılımınız için teşekkür ederiz. Bir sonraki webinarda görüşmek üzere.
Kendinize iyi bakın.